All right, our last speaker, Matej Perovnik, we will see the video in a second. I would say there was the neuroimaging statistics workshop before here, and the Keith Worsley lecture was given by Stephen Strotter, who's basically he's retired, but he came specifically to give the talk there, and he specifically mentioned the method that Matej used and is showing. So what we saw here was validity in the biological realm, so where it comes from, in the statistical realm, we saw too in that direction, that now we're gonna see something on the more clinical, like validation on the clinical realm, if you will. And the method that was used, that was originated in the 90s, I think, but when I remember correctly, was basically based on PET, or used on PET, and it's a PCA-based way of finding specific individual numbers that we can use that are associated with disease and other measures. And I think it's an old measure, but it seems very robust. And I think it's, as we said today, there's a lot of knowledge that, given that we have the huge computational capacity right now, we should still look at what's reliable and what is we can use other like robust measures for other types. So please let us watch the really nice talk.
Good afternoon, everybody. My name is Matej Perovnik, and I'm a neurology resident and a postdoctoral researcher at the University Medical Center Ljubljana in Slovenia. First, let me express my gratitude to the organizers of the symposium and of the conference for giving me the opportunity to deliver this talk today. Secondly, I also want to express my regret that I am not there in person, even though it looks like from my background that I'm here in Brisbane, but this is not the case. Because due to travel difficulties, I was unable to come to Australia. However, in the next 15 minutes, I'll present this overview talk about metabolic brain networks and the evaluation of patients with neurodegenerative disorders, rigorous validation done right. I have no disclosures. Once a biomarker is identified, it can conceptually be thought to go through a different level of evidence before it is applied to the clinical practice. Initially, we start by biomarker development, where a certain biomarker is observed in one sample, maybe with a cross-validation in a statistical manner. Then we continue with a prospective validation, new samples, direct close replications. Furthermore, if successful, then the biomarker undergoes generalization with replication across multiple labs or scanners, and finally, in a large scale diverse population. This is a hypothetical and a bit simplified overview of biomarker validation, but I think it serves a very good framework to show how one such method has been developed and applied at various levels for the analysis of brain imaging. More specifically, I will talk about the scaled subprofile model, which is based on principal component analysis, or SSMPCI abbreviated. And this method was initially and mainly used for the analysis of FDG PET-CT scans. This is a method that has been developed in the 80s, and it's routinely used in clinical practice, more in oncology, but very much so also in neurology as well. Scans are usually evaluated visually with some help of, let's say, simple mass univariate analysis. However, the PCI is a multivariate analysis, and to present it schematically how this method is devised, first the scans are obtained, obviously, then spatially normalized and smoothed. Then, after we obtain a sufficient number of control and diseased individuals, we organize this data in a subject by voxel or region of interest data matrix, extract the gray matter by thresholding or masking. We logarithmically transform the data, double center by subtracting the row and column means, and then a principal component analysis is applied to obtain subject score vectors and associated eigenvalues and principal components. After these top principal components, or a multiple of them in combination, are retained, and they represent a disease-related network, as shown here. Importantly, by applying a simple multiplication of a raw vector of a topography and patient scan, we can obtain a scalar or so-called subject score, which can then be used in clinical practice. Going back in time all the way to the early 90s, we can see the first example of this method being applied in a group of 20 healthy volunteers and 22 idiopathic PD patients. Then a topography, this is a region analysis, was identified, and it was shown that healthy individuals had lower expression, lower scores than PD individuals. But this is just one sample, the initial development. In the next step, years down the road, still at region level, 4 sites were included. Again, healthy volunteers and idiopathic patients with Parkinson's disease. And it was shown that this topography can be transferred from one sample to the second, third, and fourth sample. Then, also on a voxel-wise level. The patterns are similar across samples, and here, for the first time, we can see that this pattern is not only useful for distinguishing controls for the diseased individuals with Parkinson's disease, but also diseased individuals with Parkinson's disease from patients with dystonia, which can sometimes look similar clinically. Furthermore, an important step in the validation procedure is a so-called test-retest procedure in which we see if the biomarker is still present after some time have passed. Once again, here in this study, the PDRP was derived, now using 33 in diseased individuals, 33 healthy controls. It was validated in an independent cohort. And then comparing 2 different modalities, which can, on the left, we have data from the H215O PET, so radioactive water PET, which enables within session imaging. And it is shown by high ICC values that PDRP topography is present within 1 hour. So at the time of the injection and 1 hour after the injection, the topography will remain stable. And this is same. This is seen similarly in between session using FDG PET imaging. More recently, also functional FDG PET imaging approaches have been developed. So this is maybe something that needs to be done in the future, also for PDRP to see how stable topography is with the functional FDG PET. However, this is data from almost 20 years ago, and we can see that after 1 day and after 2 months, the topography is stable. And even more so, the expression scores from PDRP, which is a metabolic brain pattern, are highly correlated with the PDRP expression scores from the perfusion PET. Then the next step is replication across labs, scanners, and reconstruction algorithms. This was done in different studies. One such example from my home institution is shown here, where PDRP topography was derived from a Slovenian population of patients with Parkinson's disease and healthy controls. Here also differentiating them from patients with atypical Parkinsonian syndromes. And it was shown that the region weights of one and the other pattern correlated closely, and even more so the expression scores. Then we need to ask ourselves, how do the various reconstruction algorithms or scanners affect on the PDRP topography or the PDRP expression scores, especially if we want to use a pattern identified in one center in scans from a different center. It was shown in this work here that this stays remarkably similar even if different reconstruction algorithms are used. To summarize this part, in PDRP, a generalization level of biomarker validation was achieved, but also replicating the pattern in various new samples worldwide. As a matter of fact, in more than a dozen samples so far. And more recently, it was shown that PRP expression increases longitudinally over years, and that the topography can be also observed in the resting state fMRI. What about clinical utility? Here again, as shown in the introduction, it is important to bear in mind that it's not just topography, but we can also calculate subject scores per individual patient. And for the differential diagnosis, this means that after we obtain FDG PET scan from an individual, that these scans undergo pre-processing, we can calculate or evaluate various disease-related patterns on our networks. One such as Parkinson's disease-related pattern as presented, but also from other atypical Parkinsonian syndromes. And by calculating the expression of these patterns, and with the use of a simple logistic regression algorithm, which, by the way, was also derived from one population and then independently validated in 3 different populations so far, we can calculate a high probability that this patient also has Parkinson's disease. But you'll ask, how do we know this is still clinically diagnosed disease, which can be unreliable? Unfortunately for the patient, fortunately for us, the patient died, and the neuropathological exam showed characteristic signs of Parkinson's disease. Applying this method, SSM-PCA, to data from other neurodegenerative disorders such as dementia with Lewy bodies, Alzheimer's disease, frontotemporal dementia, and aforementioned atypical Parkinsonian syndromes, other patterns have been developed, identified, and validated so far. And showcasing here a large cohort of a pooled sample of more than 1,200 FDG PET scans from various sites shows that this is highly replicable across diseases, and that each disease is characterized by its own disease-related characteristic pattern. Another clinical utility is tracking disease progression and prognosis, as shown here. This is the example from Alzheimer's disease. We can see that those individuals that remain stable at cognitively normal or mild cognitive impairment state does not show elevation over time, but those that convert, for example, from mild cognitive impairment to dementia, show steady longitudinal increases in pattern expression scores. And this was also observed using PDRP and the early stage of PD, that is REM behavior sleep disorder, where this can be used to predict progression from RBD to Parkinson's disease. And in Parkinson's disease, it can be used to track survival time. So higher scores are related to shorter survival. Lastly, and briefly, let me just share you an example how this is being put forward in other disorders. And one such example is dementia with Lewy bodies. This is a disorder that is in between, conceptually thought to be in between Parkinson's disease and Alzheimer's dementia. And for the DLB, we have identified such a pattern, shown that it is present in the identification and validation cohort. This pattern has been also observed in other independent populations from Japan, China, and the Netherlands. And in our further work, we've shown that the pattern identified in one population, in this instance in Slovenia, is present obviously in the identification, internal validation cohort, and the 2 external testing cohorts from a single center and a multi-center sites. The scores correlated modestly with motor and cognitive impairment. But even more important, they were observed to be elevated in a pre-dementia state, that is mild cognitive impairment, as shown here on the right. In the middle, I show data from individuals with mild cognitive impairment, one with concomitant alpha-synuclein copathology, which is characteristic for Lewy body disorders. And we can see that this copathology affects and is in turn reflected in higher expression of DLBRP scores. Longitudinally, again, as shown for PDRP or DLBRP, we observe longitudinal increases in this pattern across years. To summarize and to conclude, SSMPCA is a well-established method for identification of disease-related patterns and networks. And here I just provided a glimpse of more than 30 years of research that showed that disease-related patterns can be used for diagnosis, differential diagnosis, tracking disease progression, following treatment response, and prognosis. And the recognition of this method is now also reflected in the latest SNMMI and EANM guidelines. And this is now named as one of the semi-quantitative analytical methods to be used in clinical practice. And as a side note, we are currently testing how this translates into higher diagnostic accuracy in everyday clinical practice. That means that in the middle, I show data from individuals with mild cognitive impairment. FDG PET scan is evaluated visually, but also with the SSMPCA algorithm to calculate the disease-related pattern scores. Lastly, PDRP is one of the PD biomarkers now in the novel biological classification called Synergy Scheme, also showcasing how a rigorous validation through decades of work can lead to real-world translation in clinical practice. I would like to acknowledge all my collaborators worldwide and thank you for your attention.
So I would say if you have any questions, write Matej, and I think it's a very elegant way of showing that simple methods can work as well or in a different way sometimes than the more complex methods. So we should really look at all the scales of methods and data. And there are biomarkers. Let's see. All right, thank you for your attention. Let's thank again for the speakers who were here, the speakers that were not here, and the organizers, where we only just were here. So thank you, and grab a coffee, and see you back in the keynote later.